Hello guys, we are back again, and this is once again Dr. D talking to you about alternate pathways of carbon dioxide fixation. We are going to look at C4 and CAM plants. Alright, let's look at C3 photosynthesis first, what we've already done before. So here, as you can see, that through the light reaction using photosystem 1 and photosystem 2, you have produced ATP and NADPH. Alright, now comes the second part, that is the Calvin cycle. For Calvin cycle, carbon dioxide is taken in, grabbed by ribulose 1,5-bisphosphate to form a 3-carbon compound. Ribulose 1,5-bisphosphate is a 5-carbon molecule, picks up 1 carbon dioxide, becomes a 6-carbon molecule, which immediately breaks up into 2 3-carbon molecules, 3-phosphoglycerate, which later on produces the pGal or G3P, that is glyceraldehyde 3-phosphate, which produces your sugars and amino acids and starch for storage, etc. This is all fine and dandy, but what if it is very, very hot? One second, let me draw a leaf in here, okay? So here's my leaf, mesophyll, and we know that there are holes through which the carbon dioxide as well as oxygen, etc. can enter. So through the stomata, these are openings. Oxygen as well as carbon dioxide are entering the leaf. But at the same time, through these openings, water, precious water, is leaving in the form of water vapor. So what happens is, this system will work fine when it is nice and humid. On a hot day, these stomatas have to close so as to conserve water. So once it closes to conserve water, what is going to happen? Well, we are going to use up all the carbon dioxide, and the environment in this leaf, inside the leaf right here, will become very, very oxygen rich. Okay? In that case, what happens is something very bad happens, and that's called photorespiration. In other words, this ribulose 1,5-bisphosphate, instead of grabbing carbon dioxide in the presence of this enzyme rubisco, now starts grabbing oxygen. And it makes products that are not needed, that will in fact harm or decrease photosynthesis, which is not good. I repeat, on a hot day, stomata closes. Stomata are openings in the leaf. If stomata close, remember, the oxygen and the carbon dioxide remain inside the leaf. So in this way, the plant is preventing loss of water. But as carbon dioxide, more and more carbon dioxide is being used up for photosynthesis, and more oxygen is being released, inside the leaf, you are producing an environment which is rich in oxygen. In that case, ribulose 1,5-bisphosphate starts grabbing oxygen instead of carbon dioxide, which is not good, which is bad. And that is what photorespiration is. This causes a decrease in photosynthesis, and you do not want a decrease in photosynthesis, obviously. So here we come across C4 plants. But first of all, let's see the anatomical differences between a C3 and a C4. If you see a C3 plant, here you are seeing the bundle sheath, and here you are seeing the mesophyll cells. These are the mesophyll cells. These are also the mesophyll cells. These are rich in, as you can see, chloroplast, where photosynthesis is taking place. So all these cells, whether it's the bundle sheath or the mesophyll cells, have an equal access to the carbon dioxide and oxygen, which is entering through these openings, or the stomata or the stoma. So now another variation are the C4 plants. When in C4 plants, I will tell you why they're called C4. They are in a bit, but first let's see the anatomy. If you notice, the mesophyll cells form like a cylinder. They are tightly packed, and inside this ring of mesophyll cells are the bundle sheath cells. So the bundle sheath cells are separated by the mesophyll cells from the rest of the environment, which out here is going to contain carbon dioxide as well as oxygen. So let's see, with C4, what is the difference? In C4 plants, carbon dioxide enters, just like this before, it enters through the stomata. And when it enters through the stomata, the first product that it is grabbed not by ribulose 1,5-bisphosphate, it is actually grabbed by PEP, which is a 3-carbon compound, which is actually phosphoenol pyruvate. As soon as this 3-carbon picks up this carbon dioxide, it becomes a 4-carbon molecule, oxaloacetate. 3 plus 1 will become 4. The enzyme that speeds up this reaction is phosphoenol pyruvate carboxylase. So oxaloacetate forms malate, which is again a 4-carbon compound in the mesophyll cells. We are here. These are the mesophyll cells. Now what happens is malate enters the inner cells, which are the bundle sheath cells inside here. And in the bundle sheath cells, the malate, which is a 4-carbon, breaks up into 3-carbon and carbon dioxide. This carbon dioxide is now grabbed by ribulose 1,5-bisphosphate, and the regular Kelvin cycle occurs. You end up with pGal, which now can be converted into sugar. There, the oxygen, we are separating this ribulose 1,5-bisphosphate, which is here, from the oxygen, which is outside here. So really, if there's lots and lots of oxygen, it doesn't matter, because now, unlike the ribulose 1,5-bisphosphate, which could also pick up oxygen, this PEP is very fussy. It's very stringent. It will ignore the oxygen and only grab carbon dioxide. So I repeat, here, this C4 plants are different from C3 plants because, first of all, the anatomy is totally different. The mesophyll cells form a ring around the bundle sheath. In the mesophyll cells, carbon dioxide is picked up by a 3-carbon molecule, that's phosphoenol pyruvate, to form a 4-carbon atom, which is oxaloacetate. Because it's a 4-carbon first carbon product, that's why we call C4 photosynthesis. The C4 oxaloacetate is converted into malate, which is also a C4 compound. That means there are 4 carbon atoms in the molecule. The malate enters the bundle sheath inside here, and in the bundle sheath safely now the malate is broken down into carbon dioxide and pyruvate. The carbon dioxide is now grabbed by this ribulose 1,5-bisphosphate. Any oxygen that was outside is prevented from entering and binding to the ribulose 1,5-bisphosphate. So if you see here the C3 versus C4, the carbon dioxide is first being taken up by ribulose 1,5-bisphosphate, and you're getting a C3 compound, and that is why that's a phosphoglycerate, and that's why this is called a C3 plant. Here, the mesophyll cells grab carbon dioxide, form a C4 compound, which comes inside the bundle sheath, breaks down to carbon dioxide and a C3 compound, and this carbon dioxide is picked up by ribulose 1,5-bisphosphate, and you'll get the regular Kelvin cycle. This is great for C4 plants. C4 plants are, of course, your corn, your wheat. These can survive in the hot, arid environment. But then we have another method of carbon dioxide fixation, which is called the CAM pathway. In a CAM plant, which are like the pineapple plant, these plants are found in very, very hot, arid places. So here, the stomata is only open at night. So when the stomata is open, the carbon dioxide enters and it is picked up by phosphoenol pyruvate again to make malic acid. The malic acid is going to sit here in the vacuole stored up during the night time. During the daytime, the stomata is closed. So this is closed during the daytime. And here, so no carbon dioxide enters, etc. Now, as in the daytime, this 4-carbon, that's a malic acid, breaks up into carbon dioxide and pyruvic acid. carbon dioxide is grabbed again by the ribulose 1,5-bisphosphate, and you are going to go through the regular C3, I will not say stuff, but the Calvin cycle goes through here, and you are going to end up with your sugars as well as with actually a pGal, and here pGal can be converted into sugars, amino acids, fatty acids, fats, etc. So once again, here's a comparison of the C4 and the C3. C4, the mesophyll and the bundle sheets, two separate cells. So the first path takes place here. The second part of photosynthesis occurs here in the bundle sheet. In the cam plants, what happens is that in the night time, there's only a night and day difference. There's no anatomical separation. Everything is happening in the mesophyll cells, so there's no phase 1 and phase 2. So in the mesophyll cells during the night time, the carbon dioxide is fixed to form a 4 carbon molecule, stable molecule, and during the daytime, when the stomatas are closed, this C4 compound breaks up into your carbon dioxide as well as a 3-carbon molecule, which can go and recycle once again, and the carbon dioxide is grabbed by your ribulose 1,5-bisphosphate, Calvin cycle takes place, and you get your glucose. So I guess with this we come to our end of our presentation. I thank you for paying attention, and if there are any questions or concerns, please talk to me or call me. Thank you.